Hello everyone, and welcome to our virtual and regional Crop Scout School this year. My name is Ashley Dean, and I am an extension entomologist for field crops at Iowa State University. And I work alongside Erin Hodson, who is also an extension entomologist and professor. And we do research and extension for the Soybean Research Lab. Today, I'll be talking to you about insect identification, sampling, and management, kind of in a broad sense, and hopefully building on some general scouting principles that you may have learned in a previous video in the series. By the end of this presentation, I hope you have a good understanding of why insects are so successful and why we care about them. I'll also go over some important insect identification features, and I'll keep this kind of broad instead of telling you how to identify, say, 10 insect species, because the importance of a particular species can vary annually and also geographically. So it's more useful for you to understand identification features and then use those in conjunction with a resource guide or something like that to narrow down the species. And then we'll go over some sampling techniques and how to recognize feeding injury. And then we'll follow up with some integrated pest management guidelines that you can follow for insect management. So there are a lot of insects in the world. In fact, there are more insects than all other plants and animals combined. And there are more than 1 million species and countless others that haven't been discovered and described yet. And I like this fun fact that 1 out of every 5 animals is a beetle, because there are a lot of beetles and it's the biggest group of insects. And it's important to know that not all of those 1 million plus species are pests to humans. And in fact, it's a really small number, and even fewer of them are actually pests of field crops. So it makes it really important to be able to identify those. So why are insects so successful? There are a lot of factors working in favor of insects, one being that they are relatively small creatures, and this allows them to live in relatively niche habitats and escape predation from larger organisms. They are also multi-generational, so this means that they can go through more than one life cycle in a single year. So they can go through 2 or 3, sometimes 18 or more generations in a year. So as you can imagine, their life cycles are relatively short. They reproduce rather quickly, and those populations can grow very rapidly. They're also one of the few animals that are capable of flight, and this has all kinds of advantages. So this allows them to be able to move toward food and habitat resources. They can migrate to suitable climates, and they can go out and find mates. They are also highly adaptable, so pretty much any food resource that you can think of, there's probably an insect that will feed on it, and that's the same thing for habitats. Any habitat that you can imagine, there's probably an insect that lives there. So they're highly adaptable, and all of these together make insects very successful. So I mentioned insect life cycles previously, and so insects go through something called metamorphosis, and the most common type is complete metamorphosis. So that would be 4 life stages: egg, larva, pupa, and adult. And the larvae often undergo several molts, which are just different stages of larval development. And the major insect groups that go through complete metamorphosis are things like beetles, butterflies, and flies. And you'll notice that the larvae and adults look very different when they undergo complete metamorphosis, and they often take advantage of completely different food resources. So for example, if the larvae are predators, then the adults might be herbivores. A less common type of insect metamorphosis is incomplete. And so that means that they only go through 3 life stages, so egg, nymph, and adult. And the nymphs often look very similar to the adults, and they will usually eat and live in the same area. So for example, if the nymphs are predatory, then the adults are usually predatory as well. And some examples of major groups that undergo incomplete metamorphosis are grasshoppers, true bugs, and then aphids and hoppers. So the most important identification that you'll have to make in the field is whether something is an insect or not. And this may seem pretty obvious, but it can be kind of tricky sometimes. So what makes insects unique is that they have 3 distinct body regions: the head, the thorax, and the abdomen. On the head, insects have 2 eyes and 1 pair of antennae. The thorax is primarily used for locomotion, so moving around, and insects have 3 pairs of legs and 2 pairs of wings to help them do that. And the abdomen is primarily what houses the digestive and reproductive systems in the insect. And insects also have an exoskeleton, so they're kind of built opposite of us, where we have our skeleton on the inside. Theirs is on the outside, and it sort of acts as a suit of armor, and it's a protective suit of armor for the insect, but it also makes it really hard to move around. So insects have jointed appendages. So their legs, their antennae, their wings, they're all jointed, which makes it easier for them to move around. And this is normally around the time when I would give you a quiz and I would try to stump you with some insects that don't follow the rules. So I just wanted to make a note that it is important to know that not all insects follow these rules. So even some adult insects like flies don't follow the rules because like flies only have 1 pair of wings instead of 2. Some adults don't have any wings at all. And immature insects are the ones who break the rules the most often. So they never have wings, and sometimes they don't have eyes or legs or antennae. So it's oftentimes a little more difficult to identify those immature stages because they don't have those identifying features. So just know that for most of what we'll talk about today, we'll be talking about how to identify the adult forms of insects. So if I'm out in the field and I've identified something as an insect, the next thing that I'll look at is their wings. So most insects have these membranous wings, which are usually clear or kind of an opaque color, and they have some veins. And the veins really give structure to the wings. Some insects, like this aphid here, don't have a lot of veins, and they're relatively weak flyers, so they kind of rely on the wind to move them around. And this ranges all the way up to something like a dragonfly, which has a whole network of veins and cross veins that makes them very excellent flyers. And this picture in the lower right here is of a butterfly. And even though it doesn't look like they have membranous wings, they do, but they are just covered in these sometimes colorful scales that we often use to identify different species of butterflies and moths. Beetles are a major group of insects that you'll see out in the field, and they have these modified forewings called elytra, which are these hard coverings on their forewings. And beetles also have membranous hind wings, which you can kind of see in the bottom middle picture on this slide. But those elytra for the forewings really protect those hind wings at rest. And you can tell that something is a beetle because those elytra will meet straight down the middle of the back. And that's how you can tell that it's a beetle. So I've included a few pictures here of weevils, which aren't called beetles, but they are under the umbrella of beetles. So they're one family of beetles. And they have the same thing, the elytra that meet straight down the back. I also have a picture here of an earwig, which is definitely not a beetle, but they also have the elytra, although it's kind of hard to see in this picture. Now, the elytra of beetles can be very long, and they can kind of meet at the tip of the abdomen, or they can be really short, like this one here. But even though they're quite variable, they will all have those elytra that meet straight down the middle of the back. True bugs are another major group that you'll see out in the field, and they have these modified forewings called hemelytra, so they're sort of half and half. The top half of the forewing, it sort of resembles the elytra of beetles, but then the bottom half is that membranous wing. And people often can get true bugs and beetles confused with each other, especially for things like stink bugs, which are really hard on the back. But the way that you can tell the difference between elytra and hemelytra is that the hemelytra of true bugs will cross over the back at the bottom. So beetles have those elytra that meet straight down the middle, and true bugs have hemelytra that kind of cross over the back. Grasshoppers are probably a group that you're familiar with, but they have the modified forewings called tegmina, and they're sort of these leathery forewings that kind of are more vertical on the back, and they kind of form a tent over the back of the insect. So that's things like grasshoppers and katydids have tegmina. So if I am still not sure what I'm looking at out in the field, the next thing that I'll look at is the insect mouth parts. And this can be kind of tricky because insects move around a lot, and maybe if you don't like insects so much, you don't want to be up close and personal with them. And sometimes they're just really small, so it would be really hard to see their mouth parts. But there are different kinds, and that can help you with identification. So some insects like grasshoppers and beetles and mantids all have chewing mouth parts. So they actually have mouths that are very similar to us, where they have mandibles, except theirs move side to side, whereas ours move up and down. Some insects have a piercing sucking stylet. So things like true bugs, aphids, and mosquitoes all have a stylet that they use to extract juices from whatever it is that they're feeding on. And so you can kind of see in this picture that the stylet is sort of folded down underneath the head and the thorax on this stink bug here. And then they just kind of fold it out when they're ready to feed. The honeybee has a more advanced mouth part where they can do both chewing and lapping. So they have the mandibles, but then they also have this mouth part that's sort of like a tongue that allows them to lap up nectar. Butterflies and moths, if they have mouth parts at all, will have this siphoning mouth part that's sort of like a big straw that they can coil up underneath their head, but then uncoil to feed. And then the housefly is an example of another very specialized mouth part that's used sort of as a sponge to soak up the fluids that they would feed on. And then the next few slides are really meant to just show some variation in the different features of insects. So the antennae can sometimes help you narrow down a family or even a species if you're using some sort of a reference key or a reference guide. So you might be seeing terms like bead-like, thread-like, clubbed, filamentous, elbowed, bristly, and feathery, and those are all just ways to describe different types of antennae. Legs are also quite variable between insects, and it oftentimes just depends on what they do and sort of where they live. So things like mole crickets that spend a lot of time in the soil have digging forelegs. Aquatic insects would have legs for swimming, and mantises have grasping legs so that they can grab onto their prey to feed. And then you sort of have the basic walking, running, and jumping types of legs, and those can all help you with identification as well. So I just wanted to point out that sometimes once you've figured out that something is like a beetle, for instance, that this slide is showing, it's still easy to get confused when you're trying to narrow down a species. So it's really hard to use colors and patterns for identification because in some insects that varies a lot even within a species. And so it's important to be able to use your references to help you make identifications. So with this example, these are all lady beetles, which are generally predators and would be the good guys, but one of these is actually a plant pest, and it's the one down in the lower right-hand corner. It's called the Mexican bean beetle, and it's considered a plant pest in certain areas, not so much in Iowa, but more in southern states. And you could get that easily confused with some of these beneficial species. So it's just important to be able to recognize the differences. Here are a few more examples of insects that we oftentimes find people get confused in the field. The first one is the beanleaf beetle and the southern corn rootworm. These can oftentimes be in the same area at the exact same time. But if you know how to identify a beanleaf beetle by the triangle that's on the back behind the head, that will help you with that identification. Another example is the western corn rootworm and the striped cucumber beetle. These look very similar, and you can tell that there are differences, but they get confused pretty frequently. The difference here is that the striped cucumber beetle comes out almost a month or two before the western corn rootworm. So sometimes people will ask us if western corn rootworms are emerging in May, and it's always the case that it's the striped cucumber beetle, because western corn rootworm does not emerge until July and August. And that's sort of the same thing with the Japanese beetle and the false Japanese beetle. Japanese beetle is a plant pest, but it normally comes out a few weeks later than the false Japanese beetle. So it's important to know how to identify individual species and also kind of know something about their biology and ecology. And this is just an example of some variation that can occur within an individual species. So on the left, we have beanleaf beetle, which I talked about a little bit. They come in a lot of different colors, and they also have varying levels of spots. And so you can always identify them by that triangle on their back. And although I didn't tell you how to identify the immature forms of different insects, this is just an example of corn earworm, which comes in a variety of different colors. And if you didn't know how to identify this species, you might not think that these caterpillars were all the same species, but they are. Okay, now we're going to switch gears a little bit and talk about how to sample for insects. So just a few key points about scouting. You always want to start looking for insects before you expect them to be there. So you want to have a few zeros on the record, so that way you can know when the insect arrived to a field and then track the population over time. And then you want to continue sampling regularly. So we often say to sample every 7 to 10 days, just so that you can have an idea of how those populations are changing and recognize when populations are getting high enough that you might need to make a management decision. And you definitely don't want to avoid areas of the field that are particularly bad or particularly good. And you definitely don't want to eyeball anything. So you don't want to scout the field while you're driving down the highway, and you don't want to stand at the edge of the field and look at injury there. And the edge of the field thing is really important because a lot of species will be overwintering or be coming from grassy areas right next to the field. And so sometimes they'll move right to the edge and begin feeding. So feeding can look really severe at the edge, but then the rest of the field has nothing. So that's why we want to encourage people to try to cover the entire field. And to do this, we would say to use a defined walking pattern. So we often use this kind of W or M walking pattern. And that allows you to cover the entire field. So you'll kind of zigzag through the field, and you'll stop at multiple spots along the way. And at each spot, you'll sample multiple plants. And then you can average what you find across the whole field to make a decision for that field. So some common tools that you might need for sampling insects is something that you can take notes on. So that could be a notebook, a smartphone, or a tablet, whatever's handy, whatever works best for you. We recommend a hand lens for those smaller insects, or if you don't have particularly great eyesight, or if you want to look at some of those features like wings or mouth parts that are really small. Lastly, a sweep net is really handy for scouting for insects because they're designed to dislodge insects from plants, and then they'll fall down into the canvas net, and you can take them into a bag or a bucket or something, and you can see what you've found. And sweep nets are good for going in early in the season just to see what's out there, or it can be used for actually scouting a particular pest to make estimations of density. And here are just a few other common sampling methods. I won't go over all of them, but just know that there are a lot of ways to sample for insects. Some of the ones that we frequently use are pheromone traps for moths. We use sticky cards for corn rootworm sampling, and the most frequent one that we use is in-field plant counts. So actually going to a plant and counting an insect on individual plants. But it really comes down to making sure that you use the best sampling method for whatever insect you are targeting. So if I were sampling soybean aphid, I wouldn't want to be using a blacklight trap or a sticky trap. I would want to be in the field looking at the plants. And the whole goal of scouting for insects is to be able to estimate pest density, so something like number of insects per plant or number of insects per sweep, or plant injury, so something like percent defoliation. And like I said on the last slide, in-field plant counts and sweep nets are really the most common tools that we use for field crops. Sometimes you'll go out to the field and you won't be able to see the pest, but you might notice that something doesn't look right, and it could be an insect. So being able to recognize insect feeding and injury is really important. So we talked about those insects that have chewing mouth parts, so things like beetles and grasshoppers and caterpillars. Even if you don't see the pest there, you can tell that they were there because pieces will be missing. So they remove plant tissue, they'll girdle the stems, or they'll clip pods or something like that, but pieces will be missing, and that's how you can tell that something with chewing mouth parts was present. Feeding injury from insects that have piercing sucking mouth parts like aphids or stink bugs, and even spider mites, which are not an insect pest, but we still care about them, that feeding is usually pretty obvious because they will be feeding on the phloem, and so they'll be extracting nutrients and water, and it usually results in discoloration of the plant tissues. So the top picture here is characteristic hopper burn from potato leafhopper and alfalfa, and you can tell that something is wrong, except we oftentimes find that people get this confused with something like a nutrient deficiency or some other agronomic disorder. So it's just important to know how to distinguish those from each other. Okay, so next we'll kind of transition into using treatment thresholds to manage pests. And this is really the basis of integrated pest management, or IPM, and it's more than biological control. So usually when people hear the term IPM, they immediately think of biocontrol, but it really is using multiple proactive tactics and using them to try to suppress pest pressure, and the ultimate goal is to have sustainable crop production. So this graphic sort of shows some of the major components of a successful IPM program. And so we've talked about a few of these, right? We talked about identification and sampling, and I told you that knowing something about the behavior, life cycle, population dynamics is really important to understand the insect. But it also is understanding that not all management tactics would be economically feasible for a particular operation. So you always want to take that into account when you're making recommendations or something like that. So some of the proactive tactics that we kind of talked about, or that I mentioned, are things like cultural control. So, tillage, plant spacing, plant populations, things like that are all ways to try to control insects proactively. Host plant resistance is another very common tool for an IPM program, and that could be genetic or transgenics, but just trying to make sure that the plant cannot be colonized by a particular pest. And then natural enemies are obviously very important, and that could be you actually releasing the natural enemies into the field, but more importantly, I think it's making sure that whatever management you're doing on the farm also fosters a good habitat for those natural enemies to thrive and help control the populations of a particular pest. And obviously, pesticides are a very important component of an IPM program, but they should really be a last resort after all of those other tactics have failed to control the pest. So pesticides are useful in an IPM program, but they should be used mindfully. So one important part of an IPM program is using thresholds to make treatment decisions. And so entomologists have worked out for many pests what the lowest population density is that will cause economic damage to the crop. So that's bushels per acre being lost, yield being lost. And they set an economic threshold below that point. So it's the point at which action should be taken to avoid the economic injury level from being reached. And that's usually something like pest density or a level of plant injury. So here's a visual representation of an economic threshold. So imagine that we have a pest whose population density follows this gray line here. We would want to make an insecticide treatment after the economic threshold is exceeded, but before it reaches the economic injury level. Treating after the economic injury level is too late. So at that first red star there, yield would already be lost, and a treatment there would be too late and you wouldn't recover the cost of that insecticide application. The worst case scenario in our minds is when someone would treat at that second red star. So that would normally be if someone wasn't scouting and that was the first time that they were in the field and they're thinking, okay, well, the economic threshold is exceeded, but it hasn't reached the economic injury level. I can make an application. But in reality, that population has already peaked and populations are naturally declining. So that would be the worst case scenario to make an insecticide application. So why would we use thresholds? I kind of alluded to this, but it helps minimize input costs for the farmer when they only make insecticide applications when they need to. Making only necessary applications also helps protect beneficials and pollinators that might be out in the field. And it also helps prevent flare-ups of other insect pests. So sometimes after an insecticide application, we might find that a new pest will move in and colonize that space, and it's usually a predator-free space because an insecticide application would take out the beneficial insects in that field as well. And most importantly, using thresholds helps prolong the efficacy of the very limited insecticide options that we have available. So it helps delay genetic resistance to particular insecticides. Insects are highly adaptable, so any time that chemicals are put out into the world and insects come in contact with them is a chance for them to overcome those insecticides. So it's really important to only use them when we need to use them. So if you've decided that an insecticide application is warranted in a field, here are some important considerations for that insecticide treatment. You want to strive for 100% kill with those applications. So that means uniform coverage of the field, the right volume and pressure, and droplet size. So a lot of the insecticides that we have available are contact insecticides, which means that they actually have to contact the insect's body to be effective. You also want to be aware of pre-harvest intervals later in the season. So sometimes insect pests will move in later in the season, but you still want to make an insecticide application. So you want to read the label. Some of those labels go out to 60 days. So that means that you have to allow 60 days from the time of application until the time that you can harvest. And you just want to make sure that you have enough time for that. We also recommend leaving check strips in the field, and this will help you assess whether the insecticide product worked as you thought it should. And then, of course, continue to scout even after the application is made. We recommend going out 3 to 5 days after an insecticide application just to make sure that it did what you thought it would do. And this is where check strips often come in handy so that you can try to identify if it's a resistance issue or if something else happened, like there was an issue with the sprayer, or it was a weather situation or something like that. So just a few take-home points. Use all the references that are available to you. They have a lot of information in them on proper identification, something about the biology of the pest, how to scout for them. All that information will be available in many of the references. So with that, you want to know something about the target pest. So you want to know how to identify them. You want to know their biology, what's their life cycle. And definitely you want to make sure that you choose the correct method for sampling. And sometimes that involves being able to recognize feeding injury. So just make sure that you know how to do all of those things. And always scout early and often. So make sure that you know when the pest arrived to the field, and make sure that you understand how that pest population is changing throughout the season. So here are just a few resources that we have where you can go to get more information. Erin and I post updates to the ICM news and ICM blog pages. So sometimes we'll post scouting advisories or things that we're seeing out in the field. And then Erin has her insect extension website where you can find some useful publications. So with that, thank you for joining me today to learn about insect identification, sampling, and management. And we're happy to take any questions that you might have for us. You can find us on Twitter. Both Erin and I are on Twitter, and we can both be reached via email. So feel free to send us any questions, or if you have any pictures you want to share throughout the summer, feel free to reach out to us. Thank you.